Material scientists have created a new material that performs like a cell membrane found in nature. Such a material has long been sought for applications as varied as water purification and drug delivery. Referred to as a lipid-like peptide, the material can assemble itself into a sheet thinner but more stable than a soap bubble. The assembled sheet can withstand being submerged in a variety of liquids and can even repair itself after damage. Cell membranes are amazing materials made from thin sheets of fatty molecules called lipids. They are at least 10 times thinner than an iridescent soap bubble and yet allow cells to collectively form organisms as diverse as bacteria, trees and people. Cell membranes are very selective about what they let pass through, using tiny embedded proteins as gatekeepers. Membranes replace dinks to their structure automatically and change thickness to pass signals from the outside environment to the cell's interior where most of the action is. Scientists would like to take advantage of membrane properties such as gatekeeping to make filters or signaling to make sensors. A cell membrane like material would have advantages over other thin materials such as graphene. For example, mimicking a cell membrane's efficient gatekeeping could result in water purifying membranes that don't require a lot of pressure or energy to push the water through. Synthetic molecules called peptoids have caught the interest of researchers because they are cheap, versatile and customizable. They are like natural proteins, including those that embed themselves in cell membranes, and can be designed to have very specific forms and functions. So the researchers decided to see if they could design peptides to make them more lipid-like. Lipid molecules are long and mostly straight. They have a fatty end that prefers to hang out with other fats, and a water-like end that prefers to come out of water. Because of this chemistry, lipid molecules arrange themselves with the fatty ends pointed toward each other, sandwiched between the water-loving ends pointed out. Scientists call this a lipid bilayer, essentially a sheet that envelopes the contents of a cell. Proteins or carbohydrate molecules embed themselves in the membranous sheet. Inspired by this, the researchers designed peptides in which each base peptide was a long molecule with one end water-loving and the other end fat-loving. They chose chemical features that they hoped would encourage the individual molecules to pack together. They examined the resulting structures using a variety of analysis methods. The team found that after putting the lipid-like peptides into a liquid solution, the molecules spontaneously crystallized and formed what the scientists call nano membranes, straight-edged sheets as thin as cell membranes, floating in the beaker. These nanomembranes maintain their structure in water or alcohol at different temperatures in solutions with high or low pH or high concentrations of salts, a feat that few cell membranes could accomplish. To better understand the nanomembranes, the team simulated how single peptide molecules interacted with each other using molecular dynamics software. The simulated peptides formed a membrane reminiscent of a lipid bilayer. The fat-loving ends lined up in the middle and their water-loving ends pointed outward, either above or below. To test whether their synthetic membranes had the signaling ability of cell membranes, the researchers added a touch of sodium chloride salt. Salt is involved in the last step in many signaling sequences and causes real cell membranes to thicken up the peptides did. The more salt the researchers added, the thicker the nanomembranes became, reaching about 125% of their original thickness in the range of salt concentrations they tested. Real membranes also hold proteins that have specific functions, such as ones that let water and only water through. The research group tested the ability of peptides to do so by introducing a variety of side chains. Side chains are essentially small molecules of different shapes, sizes and chemical natures attached to the longer lipid-like peptides. They tried 10 different designs. In each case, the peptides assembled into the nanomembranes with the core structure remaining intact. The team could also build a carbohydrate into nanomembrane, showing the material can be designed to have versatile functions. The team then tested the nanomembranes to see if they could repair themselves, a useful feature for membranes that could get scratched during use. After cutting slits in a membrane, they added more of the lipid-like peptides. Viewed under a microscope over the course of few hours, the scratches filled up with more peptide and the nanomembrane became complete again. Taken together, the results showed the researchers that they are on the right path to making synthetic cell membrane-like materials. However, there are still some challenges to be addressed for applications. For example, the researchers would like to better understand how the membranes form so they can make many desired sizes. According to the researchers, the next step is to build biomimetic membranes by incorporating natural membrane proteins or other synthetic water channels, such as carbon nanotubes, into these sheet matrix. The team is also looking into ways to make the peptoid membranes conductive for energy users.